Okay, this is Chapter 6, Section 6, and it's titled Meiosis and Genetic Variation. And the text that we're going to be going over here is going to be recognizing the significance of meiosis to sexual reproduction. The key concept here is going to be how independent assortment and crossing over during meiosis result in genetic diversity. So we'll be talking about what is independent assortment and what is crossing over, and how do they together result in genetic diversity. Okay. So we've been talking about meiosis, and talked about how meiosis is what the body uses to produce what are known as the gametes. So your gametes, we're referring to the sex cells. So for males, that's the sperm, and then for females, those are your eggs. Oops. Okay. During sexual reproduction, you create unique combinations of genes, and you do this through two main ways. The first way is what's known as independent assortment. So independent assortment, well, before we talk about independent assortment, let me review the stages of meiosis. So, we said that in meiosis you have two stages. You got meiosis 1, where you divide your homologous chromosomes. So homologous chromosomes, if you remember homo, that's referring to same. So these are the same chromosomes, meaning that, so right here, this green chromosome and this purple chromosome, they have the same genes. So, same genes. Okay, so those are what we're dividing in meiosis 1. Then you had meiosis 2, where you divide what were called the sister chromatids. And the sister chromatids, we're referring to these individual chromatids right here. So this arm of the chromosome right here, this red one, and this other arm, where this is one sister, and then this is another sister, and they were joined together at the centromere. So in meiosis 1, we're separating the homologous chromosomes, which would be this blue and this red. Then in meiosis 2, you end up splitting these sister chromatids at the centromere. And the splitting of the sister chromatids is also what happens in mitosis. So that's meiosis 2. And then the stages of meiosis, you get the same basic stages as you had in mitosis, where you had prophase, your metaphase, anaphase, and then your telophase and cytokinesis. So to review the stages real quick, prophase, you have the breaking down of this nuclear membrane that surrounds the nucleus. You have these chromosomes here are going to start to condense even more, and they condense so that you can more easily separate them. And then you have these centrosomes right here. These centrosomes produce these things called spindle fibers, and these spindle fibers, they are going to be what attach to the chromosomes here and help to pull them apart. So these centrosomes are going to start to migrate to opposite poles, opposite ends of the cell. And then in metaphase, the centrosomes have reached the opposite poles, and those spindle fibers have gone and attached to the chromosomes. Then your chromosomes have lined up on opposite sides of the equator. And so these are homologous chromosomes, meaning, again, they have the same genes. And so they line up in opposite pairs, or one pair is on one side of the equator, and one pair is on the other side of the equator. And in anaphase, you end up separating, anaphase 1, you end up separating those homologous chromosome pairs. Then telophase, you have the pinching of the you have the reformation of the nuclear membrane to form your nucleus. And then you have the pinching off of the cytoplasm with cytokinesis right here. So, independent assortment. Independent assortment is referring to what happens when you have these homologous chromosomes, and they're at this equator here. Notice how this green chromosome is right here, and the purple chromosome is right here. Independent assortment says that this purple chromosome could just as easily be where the green chromosome's at, and this green chromosome could be over here. Same for the red and the blue. How it was drawn here, the red is on this side and the blue is on this side, but that doesn't have to happen that way. There's no reason for the red to be here and the blue to be there. The blue could be here, the red could be there. And so think about that across the whole entire set of human chromosomes, all 23 chromosomes. You could have say this is mom's chromosome, this is dad's. You could have all these different possibilities of chromosomes being spread out throughout this equator, where one chromosome could be over here or one could be over there. Whereas if it wasn't independent, every time this green chromosome right here, and every time this purple chromosome would be on this side of the equator. But independent assortment, it's random. It could be on either side of the equator. And then you have what's known as random fertilization of gametes. So to understand that, think of this picture right here where I had mentioned that these are all the sperm trying to fertilize this egg. And whichever sperm makes it in to fertilize this egg is random. We don't know which sperm. All these sperm are slightly different. They're slightly different largely because of this independent assortment process and because of crossing over, which we'll talk about in the next slide. But you have all these different types of sperm, and this egg is different from all the other eggs. So you have random fertilization of the gametes. You have random gametes, and then you have it's random, which of these gametes come together in fertilization. And so this provides unique phenotypes. So phenotypes, to remind you what that is, phenotypes referring to the physical appearance. So physical. And the easy way to remember that is phenotype starts with a pH and physical starts with a pH. So it's referring to the physical manifestation, the physical appearance of an organism. So things like height, eye color, skin color, those would be examples of phenotype, example of physical characteristics. And so those physical characteristics, or the phenotypes, may give a reproductive advantage to some organisms, meaning that for some organisms, there might be a certain environment where being really tall or having, a certain color of fur gives you a reproductive advantage, increases the likelihood that that organism is going to be able to survive and reproduce to make more organisms. Okay, so moving on. Final thing is talking about crossing over. So crossing over, like independent assortment, is going to increase your genetic diversity. And crossing over is talking about the exchange of chromosomes, exchange of segments of chromosomes between your homologous chromosomes. And this occurs during prophase 1 of meiosis 1. And it results in new combinations of genes. So to kind of break this down, exchange, meaning you're trading, you're swapping different parts of the chromosome. So you got two homologous chromosomes here. So, this one from mom, this one from dad, or vice versa. And they exchange segments of their chromosome. So, this green chromosome gave this purple chromosome some of its DNA, and vice versa. So you can see they got green there and purple there. So imagine that, there's different genes, the genes on here are going to be slightly different than the genes on here. And that's going to create genetic diversity. So it's going to give you a new combination of genes. Okay, and this happens, another key thing is that this happens during prophase 1. So it happens in that very first stage of meiosis when those chromosomes are condensing. So if I go back a slide, so during here, as these chromosomes are condensing, what's actually happening is these homologous chromosomes, so this purple right here and this green, will start crossing over and exchanging some of their DNA, and same with this this teal and this red. Okay, and all that together, the crossing over and the independent assortment and the random fertilization of the gametes creates genetic diversity. Okay, and that's going to do it for 6.6.